Effective use of selective prebiotics and probiotics concentrates to control of enteritis in poultry caused by secondary gram-negative bacterial infection because of primary viral infection. Fowl adenovirus has become a major threat to the poultry industry in South Asia, especially in Sri Lanka. It is believed that disease prevalence is high in field conditions. This virus causes two important diseases in poultry known as inclusion body hepatitis and hepatitis hydropericardium syndrome. This viral infection results in heavy economical losses in broiler farming due to sudden mortality, poor growth, and flock uniformity. FADV transmitted horizontally through contaminated excretions and vertically via embryonated eggs from infected parent flocks. Sudden mortality usually is seen in chickens less than 6 weeks old and as young as 4 days of age. Mortality normally ranges between 2 and 40% in IBH and between 20 and 80% in HHS. Mortality rates also vary depending on the pathogenicity of the virus and infection with other viral or bacterial agents. Signs associated with diseases caused by other pathogens such as bacteria, fungi, or viruses commonly occur if birds are immunosuppressed. Flocks affected with IBH, HHS often have abrupt onset of mortality, and individual chickens may show nonspecific clinical signs such as lethargy, huddling, ruffled feathers, and yellow mucoid droppings due to excess bile acids. Mortality usually lasts for 5 days in IBH and longer in HHS. Inactivated and live vaccines are commonly used with autogenous inactivated vaccines prepared from prevalent serotypes in endemic regions worldwide. As already highlighted, FADV infection can result in secondary bacterial infections in poultry. Enteritis in poultry, particularly when caused by secondary gram-negative bacterial endotoxins, lipopolysaccharides or LPS, is a significant issue in poultry farming. These endotoxins are released from the cell walls of gram-negative bacteria like Escherichia coli and Salmonella species, which can lead to inflammation and damage to the intestinal epithelial lining, impairing gut health and leading to poor growth, reduced feed conversion, and increased mortality. Managing enteritis caused by bacterial endotoxins is crucial for maintaining the health and productivity of poultry. Mostly, the treatment option available for bacterial infections in poultry is antimicrobial therapy. Though the usage of antimicrobials is very effective, it has led to antimicrobial resistance, or AMR, in poultry, which is considered as a major problem nowadays. Treatments with non-antimicrobial compounds, such as prebiotics and probiotics, are becoming a trend now. Probiotics are defined as living microorganisms which confer health benefits to the host when administered in enough amounts. Properly dosed probiotics improve gut microbial balance, colonization resistance against infections, and immunological responses. Lactobacillus species, Streptococcus thermophilus, Enterococcus faecalis, and Bifidobacterium species are the most frequent lactic acid bacteria utilized in probiotic formulations. Probiotics aid in maintaining a healthy balance of bacteria in the gut by competitive exclusion. It is a process by which beneficial bacteria excludes potential pathogenic bacteria via competition for attachment sites in the intestine and nutrients and preventing bacterial overgrowth in the gut. On the other hand, prebiotics are non-digestible food elements which are selectively fermented by beneficial bacteria in the gut. Prebiotics can increase the quantity of LAB in the gut and aid in the competitive exclusion of pathogens from the gastrointestinal tract of birds. Prebiotics also increase the intestinal acidity, which aids in reducing infections in the gut of chickens. Prebiotics may work similarly to probiotics in supporting chicken intestinal health. Inulin, fructooligosaccharides, mannan oligosaccharides, galacto-oligosaccharides, soya oligosaccharides, xylo-oligosaccharides, pyrodextrins, isomalto-oligosaccharides, and lactulose are the most utilized prebiotics in poultry. The combined usage of probiotics and prebiotics during the secondary bacterial infections after viral infections such as IBH and HHS may enhance the gut health of poultry, leading to a low mortality rate and enhancing their growth. Effective control of poultry salmonellosis using probiotic feed supplement enriched with selective prebiotics. Salmonella species is a gram-negative, non-spore-forming bacillus that belongs to the Enterobacteriaceae family. It is also a facultative, anaerobic, intracellular bacillus. Most Salmonella typically grows at temperatures ranging from 5 to 45 degrees Celsius, with the ideal temperature being between 35 to 37 degrees and at the optimum pH range of 6.5 and 7.5. Some strains can even grow at a pH as low as 3.7. Salmonella is sensitive to salt concentrations ranging from 0.5 to 5% and survives in environments with higher water activity ranging from 0.96 to 0.99. They can also survive in low-moisture food products for a long duration. The severity of Salmonella infection varies according to many factors, including host age, host immunity, the presence of co-infections, environmental stress, managerial characteristics, and infective dose. Older birds, for instance, tend to be less susceptible to salmonellosis, even with concentrations of 10 to the power 6 colony-forming units per milliliter of Salmonella typhimurium. During the production cycle, poultry can become infected with Salmonella species through various routes, including contact with carrier animals like rodents, cats, and insects. Contaminated poultry feed, litter, water, and aerosol transmission also contribute to the transmission of Salmonella species. The enteric pathogen Salmonella species reaches the intestine via oral ingestion, horizontal transmission from contaminated environments, feed, and water. Even a very low infective dose of Salmonella enteritidis, as low as 1 to 5 bacteria cells, can lead to infection in day-old chicks. Both young and adult birds are infected by the invasive Salmonella enteritidis. Young birds tend to develop a systemic disease with high mortality rates, whereas adult birds can remain asymptomatic carriers. The infection dose plays a critical role in developing clinical signs. Clinical salmonellosis is more likely to develop in young birds infected with high doses of Salmonella enteritidis. Prebiotics are defined as a substrate that is selectively utilized by host microorganisms, conferring a health benefit. Prebiotics can increase the quantity of LAB in the gut, which aids in the competitive exclusion of pathogens from the gastrointestinal tract of birds. The increased intestinal acidity caused by prebiotics also helps to reduce infections in the gut. Prebiotics can also boost the immunological response, resulting in faster infection clearance. Prebiotics directly interact with gut immune cells or indirectly interact with immune cells via preferred colonization of beneficial bacteria and microbial metabolites. Prebiotics may work similarly to probiotics in supporting chicken intestinal health. Inulin, fructooligosaccharides, mannan oligosaccharides, galacto-oligosaccharides, soya oligosaccharides, xylo-oligosaccharides, pyrodextrins, isomalto oligosaccharides, and lactulose are the most utilized prebiotics in poultry. Probiotics are defined as living microorganisms which confer health benefits to the host when administered in enough amounts. Properly dosed probiotics improve gut microbial balance, colonization resistance against infections, and immunological responses. Inclusion of probiotics as dietary supplement in poultry provides beneficial effects. Some probiotic bacteria, alone or combined, have been used to prevent or control Salmonella infections during poultry production. Lactobacillus species, Streptococcus thermophilus, Enterococcus fecalis, and Bifidobacterium species are the most frequent lactic acid bacteria utilized in probiotic formulations. The mechanisms of action of probiotics include maintaining a healthy balance of bacteria in the gut by competitive exclusion. It is a process where beneficial bacteria excludes potential pathogenic bacteria via competition for attachment sites in the intestine and nutrients, and thereby prevents bacterial overgrowth in the gut. Other actions of probiotics include modulation of the immune system by balancing pro- and anti-inflammatory cytokines, production of antioxidants, and improving barrier integrity. Synbiotics consist of a combination of prebiotics and probiotics, which facilitates the implantation and survival of probiotics into the gastrointestinal tract due to their synergistic relationship. According to available reports, symbiotic-based strategies trigger some mechanisms involved in inhibiting Salmonella species or lessen the clinical signs caused by Salmonella species infection. These mechanisms may induce changes in the gastrointestinal microbiota composition, histopathological modifications in the intestine, and binding to a variety of gram-negative organisms.